the best thing about this job is that like I get older, but everyone I work with stays the same age. And you get to see how the next generation is changing and how educated they are and how much information they have. I think for them, it's like, yeah, of course you should have both of these things. But what I try to do when I teach classes is explain to them like, okay, let's say you don't care. This isn't something that like is you're passionate about. Let's talk about how studying sex differences can actually be used as a mechanism to understand fundamental aspects of biology.
My guest today is Erin Calipari. She's currently an associate professor in the Department of Pharmacology at Vanderbilt University. Her work focuses on understanding the neural circuits that govern motivated behaviors with particular attention to how these circuits are altered in addiction and psychiatric disorders. Dr. Calipari is known for integrating behavioral, molecular, and circuit-level data to create comprehensive models of brain function. Over her career, she has published over 95 peer-reviewed research articles and reviews on these topics. Welcome to Quantum Potential, a podcast dedicated to curiosity and discovery. My name is Cybele Raver, and I serve as the Provost of Vanderbilt University. As a leader in higher education and as a social scientist, I believe that we can transform higher education for the 21st century. At my core, I am wildly curious about other people's work, whether it's in science, the social sciences, the humanities, or the arts. This podcast gives us the opportunity to break open new ideas through discovery and creative expression. So one big piece of news, and I'm so excited, is that Calipari has recently been named as the new director of Vanderbilt's internationally renowned Center for Addiction Research. And through cutting-edge biomedical research at the intersection of systems neuroscience, drug development, engineering, computer science, and more, Vanderbilt Center for Addiction Research is making a difference in the struggle against drug addiction. In her role as director, Calipari is leading a large group of faculty, students, and staff as they work together to understand the brain's responses to novelty and reward and the factors that place some of us at greater risk for addiction. So welcome, Erin. I'm so excited, and thank you so much for all that you do, both in science and in terms of the impact of your work on the wider world. It's really exciting to have you.
Yeah, I'm excited to be here.
So one of the things that I could talk about, I mean, there are a million things that we could start with, but it would be really great if you could just help us by zooming out a little bit to help us understand, what does learning have to do with drug addiction? This Such an expert in both.
This is so funny because I always ask my lab, why should someone care, right? We get in the weeds of science and we're doing these really nuanced experiments. And I always ask them before we write the paper, why should someone care? When people think about addiction, I think the first thing they think about is just the drug itself. But lots of people take drug and they don't develop a substance use disorder. And so when you start to look at addiction at its core, it's really a dysregulation in how an individual learns. So, when you're thinking about learning, you don't just learn about the drug experience, but you learn about all the things associated with that drug experience. You learn about cues, things like the bar you were in, where you were when you took the drug, who you took the drug with, and all of those things become triggers for relapse. And so then you have that aspect of learning, and then you have this other aspect with, comparing drugs to other stimuli. So when you have substance use disorder, you're taking drug in the face of negative consequences, other alternative rewards. And so it's really this dysregulation of how we make decisions about drugs relative to everything else in our lives.
So interesting. I mean, I was just listening to a podcast from a British group called 1 Year No Beer. And they were talking about how when they go into a bar, when one of the co-founders goes into a bar, he goes in with the intention of not ordering a beer. And he thinks he's going to order a sparkling water. But the minute he stands up next to the edge of the bar, out from his mouth comes, I'll have a pint.
Yeah.
Right? So that idea of cues and learning makes so much sense.
And how we make decisions, your brain is kind of created to really efficiently do things. So, doing things out of habit actually is really a good way for us to automate things. The problem is when the thing that becomes automated is drug use, now you have an issue with how do we rewire these? How do we learn? What factors are influencing that? And that's really what we study.
So interesting. And so that's where you're talking really about habituation and novelty, right? Really cool. So one of the things that I loved about your new paper in Nature Neuroscience is that you really focus on the role of dopamine signaling in the nucleus accumbens to better understand the neural processes that underlie learning. Yeah. I think because you're going to take us eventually to how that connects to addiction. But first, you're really breaking down the process of learning. So just give us a quick primer, 2 minutes, about what do we want to know about the nucleus accumbens? Yeah. It's a tiny portion of the brain, right?
Yeah, so it's my favorite brain region. I always tell my neuroscience students, you have to have a favorite brain region. Mine's the nucleus accumbens. So it's a brain region that's really tied to reward. If something is valuable to you in some ways, it's a region that's very active. It's something involved in kind of linking cues and then outcomes. So you learn that something in your environment means something and you do something in response to that. It's really at the key. It's really central to that. And it's a brain region that we know is really important for just reinforcement in general. Will you do something over and over again? And it's something that's very highly dysregulated in substance use disorder. So it's kind of key to this kind of limbic system of, will you do it? Is it good? Is it bad? Should I do it again?
Cool. So is that the section of the brain that's being recruited by TikTok?
Yes. Actually, yes. And so this is, it's funny you brought this up. So a lot of these social media, all these things are really engaging these same circuits in the brain that respond to things that are good, things that make us do things again. And so you get a lot of activation in these limbic structures just by how these social media apps are really engaging you to interact with them. And so there's a lot of overlap in the circuits engaged by drugs, food, rewards, and social media, which I think is an interesting
cool.
thing.
Okay, so we'll get there in a minute. So, this little part of the brain starts firing away. And tell me how dopamine plays a key role there.
Yeah, so one of the kind of earliest things about drugs of abuse is people for a long time were kind of like, what is the thing that makes us want to do them over and over again? And so drugs of abuse, all of them, you think of opioids, you think of stimulants, you think of nicotine, alcohol. Every one of these drugs increases dopamine in this brain region. And so, in a normal situation, dopamine is increased in your brain in the same region in response to things like food, pleasurable experiences, and drugs do this pharmacologically. And so it's really that key site of those initial drug actions that are really important for motivated behaviors, but it's also a key site that really helps us identify if things are important to us or not. And so that's not just relevant to drugs, that's also relevant to any behavior, right? You put your hand on a hot stove, you learn very quickly, do not do that again. The nucleus accumbens is at the center of kind of that kind of adaptive learning. Is it important? Should I do it again? Yes or no? Should I pay attention to it?
Got it. And I really appreciate you inserting the hand on the hot stove. So it could be super pleasurable. I could be out completely vibing on the green plant life when I'm having a run and soaking it all in. And that could be really rewarding. Or alternately, I could be facing something scary or painful.
Yeah. Well, and I think, a lot of the work in my lab when I started here at Vanderbilt was really focused on understanding how dopamine is involved in reward, because that's really how we think of it. We think of it as this reward molecule, but our really early work started to realize that dopamine is not just increased by rewards. It's also increased by things that are not pleasurable. And so what we were finding is that dopamine goes up anytime something is important and something you should pay attention to. And so that could be good, bad, just something new that you hadn't noticed before. And so it's really there to kind of grab our attention and help us decide, should I do that again? Should I change my behavior? Is everything okay?
Totally clear from your papers. And what I love about your papers is that you do that in a really intense but very elegantly, you're like, oh, nothing to see here, just, hold my beer and watch this, or like hold my, just amazing, because you block and tackle each possible other alternative hypothesis. And it sounds like you're going up against a really foundational theory, right? I remember the Rescorla-Wagner model, right? Which was really that learning is really based on reward and based on trying to close the gap between whether I get the reward today or whether I'm going to get the reward if I learn something more, right? Yes. And you take that on in this like, oh, just checking. But in fact, I feel like there's this real amazing punch, counterpunch, energy to the way in which you really work through every single counter hypothesis. Am I making that up?
No, you're bringing back memories of my early years. I think that when I started in this space, that goal wasn't to be like, oh, dopamine doesn't do this. It was this. We had just gotten some new tools, which we can talk about. And I think that let us look with unprecedented resolution into these kind of very fast dopamine signals. And we were just like, let's see what happens. And when we started looking at the data, we were like, oh, this looks a little different than what we thought before. And so the science is science, the data are the data. It's when you're going up against a theory that people really like, because it's simple, it explains a lot of things. It's very challenging in a way, but challenging sounds bad. It's exciting, but it means that the burden of proof is basically, again, like you have to show
you're the
every
underdog.
Alternative. And honestly, I think on some level, when I was pre-tenure, I think I was like, oh my gosh, am I ever going to make tenure? But looking back, it really held my science to a standard that I'm proud of, because it wasn't just like, oh, here's a cool observation. It's here's a cool observation. This is what we think. And these are all the possible alternatives. And we did it being open to being wrong. Like, we weren't doing it to prove we were right. We were doing it to really systematically say, is it X or is it Y? And I think that's the most fun way to do so. I mean, that's the cool thing about academia, is that you're just trying to find out the truth. And that's what I find really exciting. But it is really hard when you have a theory, maybe revising it.
Very, very impressive. I mean, just in terms of really being eloquent about the power of discovery, right? That you're clearly getting evidence that some foundational theory that we've all been taught in our own early days may not actually be fully, as truth-based or as evidence-supported as your new evidence is suggesting. How is it to train students in that, especially when I think, it's not that it has a competitive feel, but it definitely has a persist until you've absolutely, gone to the mat.
It's okay. So I think it is a journey, but I think, one thing that makes Vanderbilt special is that we have students that are they have this spirit of kind of like a pioneering attitude. They are just excited to do it, right? And they're so amazing and high achievers that you look at them. And I think when we started down this path, any of these paths we've been on, because a lot of my work has been like, it's not really what you think it is. We always have a conversation at first where I'm like, okay, there's always a million ways you can go with a project. And it's like, do you want to go this easy way where it's like dopamine is up, let's see how drugs affect that. That's not bad. It's actually really important too. Or, it looks like we may have this potential to really revise how people think about things, but just so you know, this is going to be a really challenging path, but it'll be very rewarding. And the students here are always like, I want that one. I want the hard one that's going to revise how we think about things. And so it's a really great opportunity for mentorship too, where you start to explain to them, like, if people are questioning you, that's okay. Ask them what experiments they want to see that would convince them, and then do them.
I wanted to talk a little bit about that method that you were starting to discuss, this idea of optogenetics.
Yeah.
When I read this, I was like, wait.
Yeah.
So cool. So can you just read from your paper? This is just even on the first page of the Nature Neuroscience 2022.
Okay, now we gotta hope there aren't typos or mistakes as I'm reading this again for the first time. Okay. The development of genetically encoded fluorescent dopamine sensors allows for direct optical assessment of dopamine transients in vivo with high signal-to-noise ratio.
Okay, slow.
Yes.
So, optical.
Yes. Okay, so it's using light.
Okay.
So the some of the biggest advances in the systems neuroscience field, probably in the last 10 years, is our discovery of these different kinds of proteins that are sensitive to light.
Got it.
And so there's a couple kinds. There's some that are sensitive to light in the way that they basically can cause a cell to fire. So the way our brain communicates is cells fire, they transmit information from one neuron to another, and it's how we transmit signals. So we can actually use a laser to turn cells on or off with very high resolution. So I can say, in an environment, we turn on a light, but I don't want dopamine neurons to turn on then. I'm going to use a laser to turn them off. Or we can say, you know what, I'm going to do something else. I just want to turn dopamine neurons on and see what the animal does. So we basically are remote controlling model organisms.
Amazing.
The other class that we also use that I think is just as exciting is there are these proteins that fluoresce when a molecule binds to them. So they're for all kinds of things. The ones we use when dopamine is released in the brain, they bind to this protein and they emit light. So what we're able to do is actually record that light, and they do it really quickly. So now what we can do is we can turn dopamine neurons on and off, and we can use like optics to record dopamine release, but with like sub-second resolution. So second to second, we can say how much dopamine is in the brain, and we can look and say, okay, if there is a behavioral task where a cue comes on, you have to push a button and then you get a reward, we can separate out the dopamine responses to each part of that task independently. And so now you have all of this data where you
so
can—
it's real time. You basically are able to,
you were watching on a computer screen the dopamine going up and down while a task is being performed. And then you can go and say, well, if this pattern we think it does X, we can turn that off and say, so then the behavior should change in that way.
Far out.
Yeah.
Okay. So you use that tool to then be able to figure out exactly what you were saying, right? That it's not necessarily reward that is leading to learning, but it's actually novelty.
Exactly. So I think, you brought up Rescorla-Wagner and these different theories. And so one of the kind of, I'm like half psychologist, half systems neuroscientist. The learning theory field has made some really cool models. And what these models do is they predict what behavior should do in different cases. And so what you can do is say, okay, if dopamine was encoding reward, if we turn on dopamine neurons at this time, then the animal should do this. But if it's encoding novelty, it should do something else. And so we kind of used those previous models and what we thought would happen, and we designed experiments that, depending on what we did, the animal would do something different if we were right or they were right. And so what we did is we went through and we said, okay, let's just record dopamine release to novel stimuli. Novel is just something new, something you've never seen before in the environment. If dopamine is a reward signal, a random light shouldn't really do much to it. But if it's signaling that something new and important is here, regardless of whether it's good, bad, or whatever, we should see this dopamine response. And so what we found was that when we gave animals kind of irrelevant information, dopamine would go up. But as they became habituated to it, they experienced it for a long period of time, and they learned, oh, that's actually not important to me. Dopamine slowly went down. And so what we found was that dopamine was really like kind of stamping in these important, unexpected events, regardless of whether they were something good, bad, what it was just, was this new? Is it different than I expected? Should I do something?
So fascinating because, right? I mean, it makes so much sense from the perspective of this massive number of percepts coming into our brains through our visual field, our auditory field all the time. Like there's just a bajillion things hitting my brain all the time. And how is it that I'm going to learn that some cue in the world is important for me, either because something really awesome is going to happen if I go toward it or something not so good? I mean, so tell me a little bit about latent inhibition, right? This idea that we habituate could actually be a challenge in terms of learning, right?
Yeah. So this is such a good, the point you just made is so important. Like you can think of the world, in one way, there's unlimited amount of things in your environment, right? You're in a studio, there's a microphone, there's lights. You don't have to learn that this microphone doesn't mean I'll get ice cream. This microphone, you don't have to make an infinite number of relationships between things. What you do is you say, is this thing important? Does this predict something that I need to pay attention to, or does it not? And if it doesn't, you can ignore it. And so the idea of latent inhibition is when you've experienced something irrelevant to you, that you learn to ignore it. And so what happens now is if you were to need to learn something new about that, it takes a little bit longer because you're not paying attention to it.
Yeah, I immediately thought of the temperature gauge light in my parents' VW Bug because it's broken and it's always been broken. It's always on. And so, of course, I totally disregard that little light, right? But like one day, I'm going to be in a car. And that light's going to go on and it's going to mean like your car is overheating. But I won't probably register.
It's such a good example though, right? Is that light normally would mean something, but then you learn over time, actually, this doesn't mean what I thought. So now I ignore it. And so this is it's a fundamental process that we use in an environment to just ignore irrelevant things and efficiently learn. And so it's the reason that it was so interesting to me is that it's actually one of the violations of those previous learning theory models. And so those models were developed to just make predictions about a cue and an outcome. And one thing they didn't do well is when a cue didn't predict an outcome and you learn to ignore it, which we know happens. And so our kind of idea was, okay, well, if dopamine is encoding these predictive learning algorithms, it should be able to be predicted that what the signal looks like by these models. But if it's actually doing something different, like signaling novelty or important things, it should actually track this behavior in a way that can't be predicted by other models. So we were, I guess, using some really niche in the weeds theories. But then when you take a step back and you say, who cares? My parents aren't academics. So they're always like, who cares? And it gives me this idea of like, why should someone care? Well, because how we learn to allocate attention is really important for how we learn. And if there's dysregulation in that, you start to get deficits in behavior adaptive learning. And you see that with a lot of disorders that are characterized by dopamine dysfunction.
Right. You mentioned Parkinson's as an example, right? And does it have anything to do with addiction, or is it really primarily in these psychiatric disorders with dopamine dependent pathways?
Yes. So the reason that I ended up in this space, so my lab is like half learning theory, half really hardcore, what do drugs do to your brain? And so some of my early work had really focused on how long-term drug use changes dopamine release in the brain. And it basically showed that across almost all drug classes we tested, that long-term drug use leads to reduced dopamine responses. So you just have less dopamine responses to kind of everything, including the drugs themselves. And my question was always, well, what does that mean to an individual? You have less of a dopamine response. What does that mean? Not just to the drug, but outside of the drug context.
Got it. So the walk in the park is no longer as green.
Exactly. And or new information to you, it doesn't update your behaviors efficiently. So why should you care? Well, because let's say you're really bad at updating behavior. That means drugs, like you learn that maybe a drug cue a bar doesn't mean you're drinking all the time anymore. It's going to take you longer to extinguish that because there's new information, but you're not processing it. It's going to cause you to not evaluate negative consequences of your outcomes because that's new, or your behavior, that's new information. So you take drugs, something bad happens, you're not going to respond to that as efficiently, and you're not going to respond to alternatives. So now you have a situation that kind of starts to explain why these dopamine deficits are causing behaviors that aren't just about drugs, they're also about their relation to other stimuli in your environment that are not drugs.
So I'm just imagining you're sort of insulated from the importance of new information.
Yeah. What did you do in the past? And it makes sense, right? That's why people who've used lots of drugs keep doing that. That remains for a very long time, but they have a hard time learning new things. And these kind of deficits in decision-making are actually very predictive of outcomes with abstinence. So people who have less deficits or fewer deficits outside of the drug context, those people actually have better outcomes long-term.
So interesting. It also makes total sense. It's something like a 12-step program where you repeat the same pieces of information and the same guiding values over and over for years, right? I can understand then a little bit why that makes sense from that perspective of the difficulty it would be you'd have to update.
Right. And you're reflecting, and those things are effective, one, because you have community that holds you accountable, but two, because you're reflecting on your actions and outcomes. You're actually intentionally, cognitively saying, I did this. Was that good? Should I do it again? Yeah. What did I learn? Should I update it? So in some ways, it's really making you pay attention to something maybe that you wouldn't have otherwise. And so then you start to think about kind of what this means, how you start, conceptualizing how you help individuals. And I think that it kind of just sheds a new light on kind of why these problems emerge and how you can start to think about addressing them.
And is there, in terms of how to address them, do you feel that it directs you towards a novel or an innovative approach?
So here's the thing that's really hard is like we do not have a treatment for addiction. We have drugs that help people take less drug or that help with overdose, but there isn't like a pill you take that cures addiction. So right now what we rely on is these kind of interventions, 12-step programs, cognitive behavioral therapy. But when you're really thinking about the behavioral aspect of what these deficits mean, I think it really does help you start to, one, humanize people and think about addiction as kind of this brain disorder that's changing how we make decisions, but also start to think about how you would start to help people in that space. And so creating a drug that reverses dopamine deficits is really challenging because it's a system you need for everything. You can't just block dopamine and say, if you block the dopamine system early on, people won't take drug, but they also they feel terrible. They wouldn't eat.
Right.
They wouldn't interact with people. So
Quality of life would be.
You're trying to figure out like what systems do we target and how do we do it? And so, I think it does start to get you towards, well, how can we fine tune this system? Are there drugs that maybe would boost it a little bit at key times? And so, we're early on there, but we have a lot of people here studying that from different angles. So it's really exciting.
I mean, I love the social impact of simply destigmatizing some of those behaviors and understanding that, learning about the consequences of one's behaviors is harder under those conditions because of what's being blunted in terms of dopamine response is really huge in terms of just opening people's understanding.
Well, and I think that's why, things like this podcast, where you really think about like, the public, they're smart people who want this kind of information. And so when you start to think about things critically, but I understand why it's hard if somebody's in your life and some of their actions have harmed you to dissociate those things from the science behind it. But I do think the first step is trying to explain to people kind of what this is and really understand that from that aspect, because when you destigmatize these disorders, then people can get help. People can you can help your peers and your colleagues and your family, and you can start to think about exactly how to intervene. And I think it's a hard first step because everyone's not always willing to listen, but like as a society, I think with the opioid epidemic, we've started to have those hard conversations, and it's been exciting to see the kind of public opinion change.
Absolutely. So amazing. So let's shift to a different tool that you've used, which I just completely was super excited to read, which is your use of economic theory and principles and measurement to look at how much an individual, in this case a rodent, is willing to pay, quote unquote, like what price are they willing to pay for cocaine?
Yeah.
Right. So tell us a little bit about this remarkable study that looks at a bunch of different things, including gender differences, or I guess sex differences. I guess mice don't have gender, but sex differences in rodents. But you are also using this really cool tool to figure out how to estimate how hard we're willing to work.
Yeah. Okay. So I again, like a lot of my stuff is really focuses on kind of behavioral measures that you can actually start to think about across species. So, one of the hard things is doing work that's really mechanistic. We talked about optogenetics and optical tools. You can't do those in humans. So how do you start to take the work in that space and actually translate it into humans and primates to really understand kind of what things are translatable and what aren't? And so my background is drug self-administration. So these tasks are actually kind of cool. What you do is you put rodents in boxes and you let them push buttons. And so the interesting thing is, just like humans, they'll take lots of drug, but you can start to make the task or the game to them harder. You can say, okay, if we make you press a singular time on this button to get drug, how much will you take? If we make it harder for you, how much will you take? It's kind of like asking a person, okay, if I gave you a candy bar for 10 cents, would you pay? You're like, yeah. $1. Okay, $5. You're like, well, it's getting kind of pricey. Then all of a sudden it's like $100 and you're like, I don't know.
Right. You have to drive 2 counties away to get the Snickers bar. You're not going to get up out of bed and drive those 2 counties unless you really want it. And that's really what we're asking is, how much do you want this? And so what you're able to do now is essentially look at the price they're willing to pay. We do it in effort. Like, how much will you press to get the amount of drug you want? And you can actually use these equations that are their economics equations to say, what's the maximum you're willing to pay for how much drug you want? And then you can look at how that differs, not just across sex, species, but you can start to compare these because you can do similar studies in humans. And so what we can do here is look at sex differences. And so in one of the big parts of my lab is really studying how biological sex and hormones influence how drugs work and how we learn about them. And so in the study, what we were doing was looking at whether hormonal cycles were predictive of how much they're willing to pay, or whether they were influencing how individuals were learning about drugs and outcomes. And so what was kind of cool was that, males and females would pay, in many cases, the same amount unless there were drug-associated stimuli present. And so this kind of gets at this
far context situation that it can influence how much you consume and how hard you're willing to work. And so we're looking at how hormones influence that. It's a kind of cool cross-disciplinary series of studies where you can look at all these things and pull from different fields.
Totally. And what I thought was fascinating about your findings was that they were really complex, right? Yeah. So I think, as I recall, there are no sex differences between the male and female rodents when there are no cues, when they have access to the drug, but they weren't basically learning about the use of the drug with a cue. But that the cue specific differences arise when the female rodents are in their estrous cycle.
Yeah. So when hormone levels are high, which is that's estrus, when you have high ovarian hormones, what it's doing is it's basically strengthening the relationship between drugs and outcomes. And so essentially those drug cues have a way larger effect on behavior. And so the why should you care? So essentially what it's showing you is that how males and females are interacting with their environment is different. And the female environmental interactions and learning are really highly dependent on ovarian hormones and kind of when they're high and how they're interacting. And I think in a normal situation, these kind of hormone effects on motivation and learning are probably really important for survival. They help you learn where food is. They help you learn about safety and really adapt behavior. But then drugs are hijacking these same systems. And so it's kind of acting on a system that's there to keep us safe and then hijacking that in a way that makes you learn that drugs are more potent drivers of behavior.
Amazing. So the question that I then have for you as a scientist, and this is just flashing on my own experience, coming up at various points in various eras, is can your audience grasp the complexity? Like, do you find that they understand the complexity of what you just said? Or do you find that they go straight to, males and females are different when it comes to drugs and, they cut to a chase that you didn't actually intend?
I think it depends. I think the question I get a lot. So some of our earlier work was looking at hormones and how they potentiate drug effects. And they're just basically showing that hormones can influence how much dopamine increases with cocaine. The first thing people ask is oral contraceptives. So if people are taking birth control, does that make them more or less likely to be addicted, right? And when you start to translate—
I would not have gone there.
It's everybody goes there. But when you start to translate the findings to humans, you have to understand that there's a complex set of factors. Why does someone take the drug in the first place? Could be wildly different. It could be to avoid stress or depression, or it could just be to be social with friends. Those are fundamentally different. And so we look at these very controlled scenarios and we say, oh, well, only under these conditions. But in humans, there's just an infinite set of scenarios. Your social life, your socioeconomic status, your environment, why you're taking drugs. Do you have injuries or, other psychiatric diseases? And so—
Or I was even just thinking maybe the somatic experience of having a high hormonal level is, one that an organism, whether it's a rodent or a human, wants to blunt.
Right.
So a female human would be like,
right,
I'm really cranky and feeling achy. And
yeah, that's actually really good. I hadn't thought about that. I've thought about this, the hormonal experience being a cue in and of itself. But I think this is the thing is we get in the weeds and then the question is, how do you bring it back out in a way that people understand? And I think these kinds of podcasts and ways where you can really talk about this at a level that's maybe scientific, but also kind of explains the nitty gritty, or just I think it's the kind of first step to really making an impact.
And what is your lab? Do they just go super into, do they go completely wild for being able to ask those kinds of questions? Are they like, whoa, too complex, please give me a clear 2x2 that I can test in a semester? I mean, you talked about everybody being super ambitious, but I would imagine questions around sex differences might be either really exciting to students or feel like, oh, this is way more than I want to take on.
I think you get both because it is really hard. How do you take something that's high hormone, low hormone times, male, female, also different developmental periods, right? Hormones change over the lifespan. And how do you make something that has a testable hypothesis that your statistics can actually pull out a difference? Yeah. And I think that we spend a lot of time in the lab designing studies to answer one question, not just how are males and females different over all of it, but to say, okay, well, let's ask a specific question. And so I think some people are really excited about that. And then some people in my lab are more excited about these other questions that maybe are even simpler. Does dopamine go up or down to this stimulus or that stimulus? Are drugs different after this experience? And so I have a good mix of both, but I think they really push each other. The people studying sex differences are looking at the people studying basic mechanisms saying, who cares? Explain to me why this is important. And then the people studying basic mechanisms are saying, whoa, that's really complex. How do you distill this down to something that's testable and clear? And I think it kind of makes the science better on both sides.
Really exciting for me to think about all that work in terms of those models and how does it translate to, again, not just how people understand the problem, but, when you're thinking about the questions that people were asking, like what's the interference or promotive effect of a birth control pill, for example. Does this enter into whether treatment should look different for women who are facing trying to reduce addictive behavior versus men? Or do you feel like, whoa, too many steps, I don't want to go there?
I think so for me to go there would be too many steps. But I do think the big thing that a lot of our studies have shown is that in the human literature, we're not collecting that data like to the extent that we should have been. And so I think in the last few years, people have started doing that to ask about menstrual cycle, have hormone data, and start to say like, well, can we see in humans, are these things really influencing behavior? Like they're influencing learning on these kind of small timescales and controlled experiments, but like how much of that variance is really the thing driving behavior? Or is it just like a minuscule thing that kind of adds together with all of our experiences to kind of push things in one way or another? And so for me, the human space is like so far out of my like experimental realm, but it's kind of allowed us to have these conversations with people who are doing epidemiology work or people actually working with clinical patients to say, these are the things we see and these are the things that we don't. Yeah. And so it helps me go back and redesign our experiments. And so people are really interested in this, but also, we're talking about how hormones affect drugs, but drug use affects your hormonal cycles as well. And so that's a big thing is like repeated drug use and how that changes hormones and how that could influence behavior, the brain, stress, anxiety. And so there is, I think, a link there that needs to be considered. And I think people are starting to consider it. There's some.
And you pointed out in a recent, just listening to a different podcast that you recorded with 2 grad students. You were, I think, at a conference. And you talked about the way in which many, many drug trials funded by NIH didn't require, if they were rodent models, for example, didn't require the inclusion of both sexes, right? And so from my perspective, I totally can see where, for many, many scientists, this means having to go back and think differently, right? Test differently.
Yeah. Yeah. And so, this was a big issue. So the NIH basically mandated it was when I was in my postdoc, so it was some time ago now, where they said, hey, we need to have both males and females. And they said, you don't have to study sex differences for every study, but you can't just do all of your studies only in males. And that's what we were doing for 100 years. Basically, only 5% of all studies in the preclinical space were even powered to be able to detect.
Wait, did you just say 5%?
5% of all the studies in that space were even adequately powered to be able to tell if you even had sex differences. And so you're going back and saying, okay, well, some of these textbook mechanisms for how the brain works were defined in males, but we know there's developmental and organizational and activational effects of hormones. And some of our work that we're working on now, which will come out at some point soon, we've submitted it, is really looking at how some of these mechanisms for dopamine regulation are just not present in intact females under certain conditions because hormones change the way receptors work. And so, you start to look at like our foundation of science and how we're using that to develop drugs and treat people. And you're saying, wait, this was all developed only in males. And so sex differences aren't like everybody's different in every way, right? But there are some nuances that affect women's health, like toxicity, metabolism. There was
just dose, right? Just thinking about the amount that,
I have some studies. I should look at the years again, but there was like a 3-year period where 10 drugs were withdrawn by the FDA and 8 out of those 10 were withdrawn for having adverse health effects in women. And so these kinds of things that you think of as maybe not, oh, well, we're just studying dopamine, we're just studying this. These things really do compound down the pipeline to change how effective. Are we targeting the right things? Are women experiencing the world differently? Do they have different stressors? Are those things influencing addiction differently? And so I think even these basic studies where you're just saying, where are the differences? How are they? How much are hormones involved? Where are they changing the brain? They seem really basic, but they have a really large impact on starting to conceptualize and think about how to include women's health in the studies that we're doing.
So powerful, right? Because you're saying, I mean, as I hear you, I don't require you to study sex differences, but I do require that if you're going to study something with federally funded tax dollars, right, or federally funded support, that you actually are powered enough statistically to detect if there's an iatrogenic or negative effect, or if there's a salutary or positive effect that we didn't anticipate.
Right. Or justify where you wouldn't. I mean, there's like, yes, if you're looking at maternal behavior, you don't need to include males in those studies. If you're looking at specific effects that are affecting specific factors that influence men, like, okay, that's fine. But most neuroscience studies and most studies in my space, there is no
the brain.
Yeah, the brain. Like what? Like we're developing all of this understanding of the brain, but only in one sex. It's a pretty large problem for starting to rewrite like all of these textbook mechanisms. How many of those are exactly the same in females? And some of them, the answer is yes, and probably many of them, the answer is yes. But you still need to know that.
Totally. And so, do your undergrads get that that's a new way to look at the world, or an important major intervention on the field, or are they, coming into the lab like, of course, we should be including, I mean, I'm curious just about how much your 18 to 21-year-old newest members of science are like setting a different standard or coming in with a different set of expectations.
The best thing about this job is that like I get older, but everyone I work with stays the same age. And you get to see how the next generation is changing and how educated they are and how much information they have. I think for them, it's like, yeah, of course you should have both of these things. But what I try to do when I teach classes is explain to them like, okay, let's say you don't care. This isn't something that like is you're passionate about. Let's talk about how studying sex differences can actually be used as a mechanism to understand fundamental aspects of biology. Like, let's say you're studying how a receptor regulates dopamine release, but in females, it doesn't. Well, now you have a biologically relevant scenario where that receptor does X and Y that you can start to do studies to manipulate. Well, in which cases where it's not there, what does behavior look like? So I try to...
You have a basically built-in knockout gene, right?
Yeah. So I think I look and I say, look, okay, this is something we need to do from like a societal standpoint, but also like think about this as an opportunity to understand how variation in neural systems and behavior influences our experience in the world. And so I think my goal is to try to get people to understand how important this is. And I think you have to meet them where they are. Like you meet some people and they say, yes, I get it. And you say, great, let me give you the tools. And other people are like, I don't care. And I'm like, let me show you some other ways that this could be used, even if that's not your key question. And so it's kind of exciting. It's a challenge for me, but it also kind of keeps me on my toes.
Absolutely. And generally, are students understanding themselves to be future scientists? Who's in your class? What are those students thinking they are in the class to learn? Yeah.
So, okay, we have a good mix of people. I think like one of the coolest things is how they're kind of going beyond these traditional paths. Like, it's like you go here, then academia. These students are rewriting kind of what things look like for them. They're collaborating across colleges. They're doing immersion projects. A lot of them, I think, come in not necessarily knowing what an academic career looks like. It's not something, if you haven't experienced it with your family, that you really get until you get to an undergrad institution where you say, oh, I can just like learn stuff all the time as my job. And so you have some of that. You have people that want to go into the medical profession, which I see this as an opportunity for me to explain to them kind of why these things are important. You have people who really want to do drug discovery. Again, like this kind of work is really critical. And then you have people who want to come up with solutions that I can't even think of for problems that we haven't encountered yet that develop, their own companies. And so you have this really diverse perspective. And as an educator, you're sitting there saying, how do I engage all of these different people? And I do think that makes my science better because it's that, who cares? why would all of these people care? And it really makes me think about my work in that context. How do I convince these people it's important? And if I can't, maybe it isn't. And so, which is sometimes sad, but like,
no, but what a scientific approach to your own.
Yeah. So I try to educate them, but then I kind of look at what they are seeing is the next thing and what they're looking to get out of this. And I say, one, I'm going to try to convince you to come over to my academic side, but I also want you to kind of help me shape what we think is important. It's fun.
It's just great. They're so lucky to have you, Erin. It's just wonderful. I think when I have a chance to step back, I immediately want to know what you really want us to have as a key takeaway. You know, if you're thinking about someone who isn't at Vanderbilt or isn't part of a university community, and they've heard you talk about this incredible role of, you know, the endocrine system and dopamine and drug use, from all of the things that you've just shared with us, what's a key thing that you want us to take away?
Yeah, that things are way more complicated than we think. I think that we as humans like very simple explanations for things. And I think in some level that's amazing, right? But I think that things are really complex. And I think thinking deeper, and you can't think deeply about everything, but about things that are important to you, I think really opens up how you approach the world. And so I think the dopamine work we do is a really good example of dopamine is a reward molecule. Well, why do we think that? And you start to see that that's more complicated. And thinking about how hormones influence that and how you can change how you learn at specific times is also interesting. And so I think, like, what is the take home? The take home is that the brain is a really complex organ that helps us learn and adapt to our environment in a way that is efficient and interesting, and that there's lots of things in our environment that can hijack that for better or worse.
Yeah. I mean, profoundly hopeful to me, right? Because in a key way, it's not just a person with a set of test tubes and a set of mice trying to figure out a special arcane pathway. You're actually trying to address the potential for, human flourishing in the face of something that I think for many of us seems really scary. The opioid epidemic seems intractable, right? But you're demonstrating all these amazing, hopeful sort of avenues that we've only just begun to pursue.
Yeah, and I think that's the interesting thing. And the fact that people are becoming more and more interested in getting accurate, factual information, I think it's just such a cool time that we're able to kind of put this out there and people actually want to engage and understand it deeper. And I think that bodes really well for human disease as well, because it means people are going to be invested in solving this problem in a way that we maybe weren't in the past.
Thank you so much, Erin, and thanks to our listeners for tuning in. We'll be back next time with another guest who's pushing the boundaries of discovery. Until then, I really want to thank Erin Calipari for her leadership and her amazing groundbreaking science. And I really want to thank every one of you for having tuned in. I'm Cybele Raver, and this is Quantum Potential.
The Quantum Potential podcast comes from Vanderbilt University. Your host is Cybele Raver. Thank you to our guest, Erin Calipari. Production by Matriarch Digital Media. For more information about quantum potential, go to vanderbilt.edu /quantum potential. Stay curious.